Hello, my name is Candace Beaver, and I am with the United States Department of Agriculture. Today I'm going to describe how we have developed a rapid portable device for the detection of amatoxins. This device uses a technology called lateral flow, which is similar to how a pregnancy test runs. And we're going to show how we've used this lateral flow immunoassay to detect toxins in mushrooms and in urine samples. There are a lot of mushrooms out there, and of the thousands that have been described, a lot of them are not edible or they have unknown edibility, they haven't even been tried, or they just taste too bitter. Some are edible, some are hallucinogenic, and some are poisonous. But there's also a lot of lookalikes. So where we have morels that have this iconic look to them, there's also something that can be mistaken for them, which is the diarmitra. There's also candy caps, which are little brown mushrooms, and maybe to an untrained eye, they look like these psilocybes or these hallucinogenic mushrooms, or like a deadly galerina, which is also a little brown mushroom. There's also amanitas, which have some choice edible mushrooms that people seek to eat. However, there's also the most deadliest mushrooms, which are some of these shown here, that look very similar to a lot of the choice edible amanitas. Of those poisonous mushrooms, they can have a range of toxicities. This chart shows in decreasing order the lethality of many of those toxins. You can see here that the chemical structure is very different for each type of mushroom toxin. And you can also see that representative species are very distinctly different. The way mushrooms are identified is based on morphological features, such as the physical shape and structure of the cap, the gills, the stipe. You can also look at the color of those too. You also need to look at the ecology. What substrate is it growing on? What season is it growing in? And then also looking at the spores. The spore print will tell you the color of the spores, but you can also use a chemical test, such as the Melsner's reagent, that can stain the spores so you can look at them under a microscope. Potassium hydroxide has been used to turn caps and the yellow color to tell them that they're a yellow staining mushroom, which can be indicative of a poisonous mushroom. There's also the Meissner-Weiland test that tests for amatoxins. When you can smear a mushroom onto newspaper with lignin in it, and then add concentrated hydrochloric acid, if the amatoxins are present, they will turn the paper blue. And then if you have access to DNA sequencing, you can sequence the mushroom to identify it down to species based on multiple genes. So what are amatoxins? Amatoxins are a class of molecules. They are the most lethal mushroom toxins, but there's not just one of them. There's actually a variety of them based on small changes in different R groups on this backbone structure. The most well studied are alpha, beta, and gamma amanitin. They are RNA polymerase II inhibitors, which essentially halt protein synthesis and thus results in cell death. The LD50 in humans is about 0.1 milligram per kilogram, and so if you can imagine that a person weighs approximately 70 kilograms or 150 pounds, they need about 7 milligrams of toxin to kill about half the people that ingest it. And in fact, one mushroom contains a lethal dose. These toxins cannot be destroyed by cooking, drying, or freezing, and they're not produced by all mushrooms, but just by some of the mushroom species. The mushroom species that make amatoxins are the most commonly known are the amanitas, such as Amanita phalloides, Amanita ocreata, and Amanita bisporidra. There are also a few other genera that have amatoxin producing species, which are Galerina marginata, as well as a conocobe is suspected to have amatoxins, and some lepiotas contain amatoxins. Amatoxins are really interesting in that they are actually made on the ribosome, which means that there is a DNA sequence to transcribe for this peptide sequence. And so you can see this peptide sequence is actually in the genome of the mushroom. The mushrooms also make typically about 0.2 milligrams per gram of toxin, 0.2 milligrams of toxin per gram of fresh mushroom, which equates to approximately, if one mushroom weighs about 100 grams, 20 milligrams of toxin. The Amanita phalloides are also known as the death caps. In a report about 15 years ago, the death caps were shown to have been native to Europe, but in the last 100 years or so have been introduced to almost every other continent on the planet, except Antarctica. More recently, I took some iNaturalist data, which is citizen science reported observations of the death caps, and I overlaid it on that original graph just to show that they are still present in a lot of the same areas and that they're still very widespread across the planet. Now, just because amatoxin-containing mushrooms are widespread doesn't mean that we will, in fact, be exposed to them. However, unfortunately, usually through accidental ingestion from misidentification of a mushroom, we humans are exposed, as well as other animals. And historically speaking, the exposures are the highest in the areas where there are more mushrooms found. More recently, in 2020, here's just a little picture of how many other exposures have occurred in certain regions, and for the most part, sometimes there's a lot of poisonings, and some recover and some do not. Now, to put mushroom-related exposures in context of other food-related poisonings, you can see here that for the most part, mushrooms actually have a higher death per exposure percentage. Now, that is to say that they have the lowest number of exposures, about 6,000 per year, compared to plants and infectious diseases such as botulism and salmonella, but it does result in more deaths per exposure. After exposure, symptoms typically take about 6 to 12 hours to manifest. Those will usually be in the form of abdominal pain, nausea, vomiting, and diarrhea. Currently, there is no FDA-approved treatment, at least not in the United States. Supportive care is what is provided to most patients, and that includes giving IV fluids and also trying to induce vomiting and give activated charcoal. If it's severe, liver transplant is your next option for just entirely removing your liver and getting a new one. In the European Union, milk thistle has been approved to be administered by IV for amatoxin poisoning cases. And for a clinician, predominantly the diagnosis is based on a history of ingesting wild mushrooms, the symptoms that were discussed earlier, as well as clinical labs showing high liver enzyme activity. One tool that we could use to diagnose amatoxin poisoning would be to detect the actual toxin. And here is a study from over 20 years ago where they showed that the concentrations of amatoxins in patients that have been exposed to amatoxin-containing mushrooms, such that the urine levels were at thousands of nanograms per mil early on after ingestion, and then they dropped off over time, and that the concentrations in plasma are shown on the right. So this suggests that if we wanted to make a diagnostic for amatoxins, that we should evaluate urine because it lasts longer in urine and that the concentrations are higher in urine, and that we need it to be at the nanogram per milliliter level. So the overall goal of this project is to develop a rapid test for amanitin, or amatoxins. We wanted to make it into a lateral flow immunoassay, which meant we had to develop reagents, which are both the antigens and the antibodies. We needed to incorporate these reagents into a rapid assay format, and then we needed to test it and validate it with samples that contain amanitin, such as mushrooms and urine. And again, because mushrooms make it at such high levels, at the milligram level, that's the detection limit we're aiming for mushrooms. And then for urine, we're looking at nanogram per mil levels. The way this test works is similar to a pregnancy test, except that it has a readout that's opposite, such that when there's 2 lines present, it means the sample is negative. When there's 1 line present, it means it's positive. The reason this happens is because on the control and test line, we have immobilized reagents already pre-set into the test strip. On the test line is our antigen, which has an amanitin surrogate attached to it. And on the control line, it's just an anti-mouse to antibody that just indicates that the test did work. Next, we have a anti-amanitin antibody that's been pre-conjugated or already conjugated to a gold nanoparticle, which is what gives everything the pink color. And those are pre-loaded onto a membrane in the bottom of the cassette. And when a solution is added, these reagents are then mobilized such that they can bind and freely react with both the test line and control line. When amanitin is present in a sample, and then it's added into a solution, it binds to the antibody, thus not allowing it to bind to the test line, and it can only bind to the control line, resulting in 1 positive line indicating that amatoxins are present. To characterize this assay, we want to determine the detection limits and how sensitive it was in different reagents. So the first thing we tried was PBS, And in phosphate buffered saline, you can see on the control line should always be present, indicating the test works. Whereas on the test line, as with increasing concentration of amanitin, the line disappears. You can also see that in the graphs on the right, where at high concentrations of toxin, you get a low pixel intensity, and for a low concentration of toxin, you start to actually see a pixel intensity indicating that line is present. With urine, there's also slightly an improved sensitivity, and that's likely due to the pH. The urine is a little bit lower pH than PBS. But all this to say that this test is easy to read by eye, and it clearly indicates the presence of 10 nanograms per mil of alpha amanitin, which is great because that succeeds in being able to test from mushrooms, the milligram level, and it also achieves the urine analysis level of nanograms per mil. Further characterization of this assay is to determine what else does it possibly bind? What does it cross-react with? And so you can see at the very top line, the alpha amanitin is our goal, our target molecule, so its cross-reactivity is 100%. One of the similar amantins is gamma amanitin, and it binds and reacts to it just as sensitively as it does alpha. This test does bind to beta a little bit less sensitively at about 0.5% cross-reactivity. And then there's also these phallotoxins that exist in Amanita phalloides and other Amanita species, and that's phalloidin and phallocidin, and those both cross-react at very, very small amounts. And then it does not cross-react with psilocybin or ibotenic acid or muscimol, which are other toxins that are found in some other wild mushrooms. And so all this to say that the assay is selective for amatoxins. As you may have noticed, the mushroom is not an aqueous solution that can be added directly to the lateral flow test strip. So I needed to figure out how to get the toxins out of the mushroom. So I looked to liquid chromatography mass spectrometry methods for how they did sample preparation, and I saw that for the most part they would grind up the mushroom tissue, extract it with a liquid extraction, and then do centrifugation and filtration. However, I found I do not need a filtration step because I already have a filter embedded in my lateral flow test strip cassette. I also thought we don't need to grind up the sample because the toxins are readily available from the tissue. So then I had to examine what kind of solvent I can use and how long should I perform the liquid extraction. And so here are the data to support how I did the sample preparation optimization. And what I did is I first did a methanol extraction on an Amanita phalloides tissue, dried tissue, and I did all three methods, bead beating and mortar and pestle grinding and hand shaking. The mortar and pestle grinding is usually done for about 60 minutes, and so that's considered the gold standard. You can see from the green bars on those samples that were extracted, we obtained approximately the same amount of alpha amanitin out of 60 minutes, 30 minutes, and 10 minutes, and the same is true with the 1 minute hand shaking, and these were detected by HPLC UV. So I thought, well, that was suitable to say that handshaking is sufficient. Then I realized, though, that methanol is not a suitable solvent for lateral flow immunoassay, nor is it for an ELISA. So I tested different solvents. I used methanol, water, phosphate buffer tween, and tris buffered saline solution with tween, and I saw that I could extract nearly a lot of the toxin and almost the same as the methanol extraction in 1 minute, and you can see that's shown by complete inhibition of the competitive ELISA. And then as the dilutions increase, the signal starts to become apparent. So here you can see that the final method that I chose to do was to shake by hand for 1 minute with just PBS solution. So here's a quick summary of the test strip method that we've developed. It selectively detects alpha, beta, and gamma. The sensitivity is about 10 nanograms per mil. It provides a qualitative readout, yes, no. It does also require a small amount of urine, 100 microliters, or a very small amount of mushroom tissue. No extractions needed for the urine sampling, but there is a quick extraction needed for mushroom samples. To utilize this test in mushroom sampling, I gained access to an herbarium and I was able to use a bunch of dried mushrooms. I had access to over 110 different specimens, which included 96 different species. So you can see some of the pictures there of different mushrooms that I tested, and you can see that's shown here is 1 through 23 23 different samples that were tested, but in all there was 110. What I'm showing is the most interesting ones, such that you can see at the test line where it does not exist, that's a positive for amatoxins. And those are the 6 that are shown in red below. And there were interesting ones such as number 8, Amanita muscaria, the red iconic mushroom there. It has other toxins in it, but it does not have amatoxins. And then you can see some edible amanitas also shown here. Amanita villosa is the picture right above to the top right of number 14, and that's an edible amanita, and it of course shows that it's negative for amatoxins. Next, we wanted to test human urine samples. And so for this validation, we compared our results to that obtained by CDC and the California Department of Public Health, and they were running the LCMS method. For this, we had the CDC blindly spike urine samples with varying amounts of alpha, beta, or gamma amanitin congeners. Sometimes they spiked them with 1 congener, sometimes 2, and sometimes all 3. And then sometimes there was none. And for the lateral flow assay, we only determined detected or not detected. And you can see that those correlate very well with the samples that did or did not have toxin present. And then for LCMS, they actually were able to get higher resolution analysis and be able to detect each individual congener. And next we wanted to test real samples. And here we were able to collaborate with a local vet who happened to see a lot of amatoxin poisonings in dogs. And she provided us with a bunch of samples, both from suspected and non-suspected cases of intoxicated dogs. We also collaborated with UC Davis, the California Animal Health and Food Safety Laboratory, who has a validated LCMS method for amatoxin detection. And typically, the vet would actually collaborate with the UC Davis Center, in that when she had suspected cases, she would send the samples to the UC Davis Center and they would analyze it for her. They would report true positives if the concentration of amatoxins was greater than 1 nanogram per mil. They reported trace results for samples that they can't quantify, but they did get a positive hit at the right elution time, as well as having a right mass volume. And then they also had non-detect if it was below their detection limit. And you can see that for the samples that were true positive, when we detected them, they were positive by LCMS. There was one sample that was a positive result for LCMS that we could not detect, and that was because their sample was about 2 nanograms per mil, which is below our detection limit, because remember our detection limit is 10 nanograms per mil, but we could not quantify the below that. And in the rest of the cases, we would expect where the trace results occurred, they are below our detection limit, so those were negative. And then also we'd have true negatives for all the rest of the samples. What's also been interesting, and just during this pandemic, we've had a couple other cases come in of amatoxin poisoning, and these are examples of the test strips that the physician would send to me when they received a case. Now, in order to identify if the diagnostic test is very accurate or efficient, we compare all of the true positive, true negative, false positive, and false negatives from different samplings. Here I did show the intoxicated dog urine samples and that the accuracy is almost 80%. I didn't show in this report, but you can see it in our paper, fortified human study where we just spiked samples blindly. The CDC spiked them and we analyzed them. And you can see that we pretty often never have false positives. And this is just to say that nothing in human and dog urine interferes with our test. However, we do sometimes have false negatives, which is that sometimes the concentrations are below our detection limit. And either that's because it's detecting beta amanitin, and LCMS can detect beta amanitin better than we can, or the concentrations are more sensitive, can be detected more sensitively by LCMS and not by our method. I'm often asked, what is the shelf life of the test strip? So to test this, we do what's called an accelerated stability study, where we subject the test strips to high elevated temperatures for a certain number of days, and those equate to a standard day. And in this case, we subjected it to 55 degrees Celsius, when room temperature is about 25 degrees Celsius, and that's what typical storage would be at. And you can see that when we added different concentrations of the target we're intending to detect, the only time we see a change in the visual score is when the blue line, where there's no toxin being added, it's just the signal intensity is starting to drop at about 35 accelerated days. The signal also starts to drop at 50 accelerated days for the 1 nanogram per mil test. So you can say that this equates to approximately a year and a half shelf life. However, it's probably also much longer than that. So here is a chart to show the comparison of amatoxin detection technologies. Meissner test is that simple hydrochloric acid test where you get a visual blue spot. You can see that it takes a couple minutes, but it also cross-reacts with psilocybin and other indoles. There's LCMS, which has higher resolution. It does take a little bit longer, but its sensitivity is great, and it does require an expensive instrument. There's also an ELISA-based method that takes an hour, has a comparable sensitivity of nanogram per mil, and it relies on a plate reader and a polyclonal antibody. And then you see the test strip, how it compares, where it's fast, it takes a couple minutes, 10 minutes. Its sensitivity is reasonable for urine analysis and for mushroom analysis, and you get a visual red band as your readout. And with that, I thank you for your attention, and also all of my collaborators, which have contributed to both finding mushrooms and also finding samples and have an interest in developing and detecting amatoxins.